Good evening, everybody. On behalf of the Center for Cold Waters Restoration, the Virginia Museum of Natural History, and the Wayne Theater, welcome to our November 16th Science Talk. Concerning the project, what you're seeing on the screen here is just a rolling slideshow. For those of you who have been at our science talks here for the last 2 years, you've seen some of these images already, but these are depicting the early concept designs for the Virginia Museum of Natural History Waynesboro campus. Just a reminder, it's a 25, almost 30,000 square foot project that will be located right here on the banks of the river and next to Constitution Park, or what will soon be the South River Preserve. This project's been approved by the General Assembly, and right now we're just waiting for some final signatures to get the initial funding to go out and put out the bid to hire an architectural engineering firm to create the detailed designs. We're really excited about this. This is a great project for Waynesboro. It's a world-class facility. As you can see from some of these exhibit designs in the building itself, it's really going to put quite a bookmark on the end of our downtown area here. We've got several groups, about 5 teams, design teams, that have started meeting to work on recommendations for different concepts. Covers everything from the exhibit design to the building and landscaping. These groups have started to talk and start developing a list of suggestions that'll be the input that's going to be provided to the A&E firm that they'll use to design the project into details. And then early into next year, January, February or so, we'll be calling a community-based meeting here in Waynesboro for the community to meet the architects as well as to start seeing the plans develop, and also give you an opportunity to provide some input into what you feel this museum should represent. My name is Len Poulin. I'm the president of the Center for Cold Waters Restoration, and have been, our organization, which is basically depicted here, is an economic, it's really an advocacy group to advocate for natural resource-based economic development. And the museum is kind of like the second project in our overall plan for redeveloping our economic future, centered around our natural resources. The fly fishing festival and the industry that it spawned here, no pun intended, kind of got that kicked off. Any case, we've got a couple board members here from the Center for Cold Waters Representative. Irby Nash is one, and Jenna and Hal Althestead, all great guys. That, we're all volunteers. The community, or this group, basically sits down to envision and to brainstorm these concepts, and we've been working with Joe in the museum here for quite some time to come up with this project. So we don't have Science Talk scheduled for December. We take the holidays off. Our next one will be January 11th. And the topic will be life underwater, aquatic insects and their feeding habits in the South River. That'll be presented by Dr. Joe Kuiper, who will be up here in a second, who's the executive director of the Virginia Museum of Natural History. On February 8th, the topic will be vernal ponds. It'll be presented by Stephen David Johnson, who is a conservation photographer and professor of visual and communication arts at EMU. This is kind of a different kind of talk, in the sense that he interprets vernal ponds through the lens of a camera, and I've seen some of the images, and they're just amazing. So if you want to learn about vernal ponds and you want to see some amazingly beautiful pictures, come to this particular science talk. It's going to be very interesting. On the 15th of March, the history of the South River fishery. That's going to be presented by our own Irby Nash, as well as Steve Reeser. This is going to talk about the fishery here in the river and how it's developed over the last 20 years, pretty much. And then going back to some older history than that. And then our last science talk is scheduled for April 12th. And the subject there is effects of climate change on Tangier Island and the efforts to make the island more energy efficient. This will be presented by another CCR board member, Dr. Jonathan Miles, who is a JMU professor of integrated science and technology, and is also the executive director of the JMU Center for Advancement of Sustainable Energy. So as you know, Tangier Island's pretty low, and as the water levels increase, there's a civilization or community there that's under threat. So we're going to talk about the impact of that. And with that, I'd like to ask Joe Kuiper to step forward. He's going to make a short presentation.
All right, thank you, Len, and good evening, everybody. We started working on this project to establish a branch of our Museum of Natural History here nearly a decade ago. It started with just local conversations with supportive people, and that led to a deep relationship between the Natural History Museum and the Center for Cold Waters Restoration. And so they have really shepherded us to this point where we're in the state funding flow. It's super critical that we got to this point. And the Board of Trustees recognizes that. So at their last meeting back in August, they actually presented a resolution to express their thanks to the people who represent the Center for Cold Waters Restoration. So Len, I'm just going to take a minute and read their resolution here. This is very formal. All right. So, from the Board of Trustees, Virginia Museum of Natural History, whereas the Center for Cold Waters Restoration, CCR, has provided invaluable assistance to the Virginia Museum of Natural History in the establishment of the VMNH Waynesboro Branch Campus, whereas CCR has connected staff and board members to VMNH supporters throughout the state, whereas the Center for Cold Waters Restoration advocated for the establishment of VMNH Waynesboro in the General Assembly in the city of Waynesboro, whereas CCR provided expertise in the development of a campus master plan and pre-planning study, whereas CCR connected the museum to community events, assisted with the development and management of an annual lecture series, and drew needed attention to the museum's work and needs. Whereas CCR has made it known it will continue to support the establishment of VMNH Waynesboro during future phases of design and fundraising. Therefore, be it resolved that the Board of Trustees of VMNH greatly expresses to the Center for Cold Waters Restoration its appreciation and thanks for a decade of expert and enthusiastic service to the museum done on this 21st day of August 2021 in Martinsville, Virginia by a unanimous vote of the board, signed by yours truly, but more importantly, Ms. Jennifer Burnett, Chair of the Board of Trustees. So on behalf of the Board of Trustees, Len, I will pass this on to you to share with your colleagues. So thank you so much.
Thank you. I actually knew this was going to happen tonight, but I didn't know it until yesterday, and I didn't realize this was happening. So many thanks to the Board of Trustees for recognizing the work. You know, Joe mentioned a decade. It's been at least that we've been working on this. And these things don't come quick, and it takes time and effort by, you know, amazing volunteers. I can't say enough about our Board of Directors. We've got some incredibly talented people, very dedicated, very insightful, that are working in the background. You don't hear much about the Center for Cold Waters Restoration, but these folks are developing a strategic plan for our community that's going to set us up for an industrial future based on natural resources. So my hat's off to the Board of Directors here, and I can't wait to bring this to our next board meeting. Okay, enough said, so let me introduce our speaker. You may have remembered, if you were here about 2 years ago, in 2019, Dr. Jess Jones had come and talked about did a presentation on mussels. The reason he came to talk there is because the DuPont Nerdist Settlement funded a project in Virginia Tech to repopulate or reseed mussels into the South River here in Waynesboro. Today, our speaker today, Brian Watson, is going to talk about that effort because he's one of the leaders of this project. So the topic is freshwater mussel restoration in Virginia and the South River watershed. Brian Watson earned a BS from the University of Tampa in marine science and biology and an MS from Virginia Tech in fisheries science, focusing on the life history of 4 federally endangered species of freshwater mussels. Afterwards, he worked for the North Carolina Wildlife Resource Commission as a non-game aquatics biologist, conducting surveys for freshwater mussels, snails, crayfish, and non-game fish across the state. Since September 2002, he has been the state Malacologist, correct? Hey, see, I didn't mess that up. There's a reason why I'm not a scientist. I can't pronounce 90% of all the stuff I see. For the Virginia Department of Wildlife Resources. By the way, in case you're wondering, the Malacology is the study of mollusks, mussels, clams, snails, squid, and etc. On October 12th, Brian was one of the scientists involved in a project to restock mussels in the South River. Students from our Waynesboro High School's environmental science class were invited to join First Lady Pam Northam in placing healthy mussels in the river. It was on Channel 29. I think the teachers in the room today, right? Kristen Torover, just say hi. Wonderful. And are some of your students here too? Were some of those folks in there seeding mussels? Maybe. Anyways, without further ado, Brian, thank you. And by the way, I'm a shrimp, so you might have to readjust this. Come on.
Thank you. Thank you for coming out this evening. Just a quick show of hands. How many people are familiar with anything about freshwater mussels? Yeah, fair number of folks. That may be because of the DuPont settlement, which I'll talk about a little bit more towards the end of my presentation, but I will go over and give a general overview of freshwater mussels, what they are, why they're important to us, what their current status is, and what we're doing to try to restore freshwater mussels across Virginia and also with the specific South River DuPont restoration project here in Waynesboro. And if anybody has any questions during the talk, please feel free to holler at me. I don't necessarily need to wait until the end. Yep, it's on now. There we go. Okay, so what is a freshwater mussel? It's a soft-bodied invertebrate with a hard shell. If any of you ever handled them, typically what you have in your hand is that hard shell, just like a saltwater clam. But inside they have the soft tissue, mainly a muscular foot that they use to burrow down in their habitat. Most of them live in streams and rivers. Some can live in lakes and ponds. They rely on a current to supply their food. They're filter feeders. They're usually feeding on detritus and algae in the water column. They have a very widespread distribution across the globe. They're on every continent except Antarctica. They're sedentary most of their life. They don't really move a long distance unless they get swept downriver during high flow events. They have a complex life cycle, which I'll get into and show you some video. That's usually kind of the cool part of looking at some of the life cycle of these guys. If you haven't seen any of those videos, usually that's kind of the wow, bang, whiz portion of the presentation when you see some of the unique and distinctive kind of life history and displays that they have to try to complete their life cycle for such what's considered a primitive organism. And they're highly diverse and a highly imperiled taxa. So just kind of go over just the basics on freshwater mussels. This is just one example. This is we got a purple warty back here. A lot of them get their names from what the mussel looks like. So you can see this one has bumps on the outside of the shell, purple nacre on the inside, so purple warty back. You can see we've got a display of some mussels up here. A lot of times they get their name from what the shell looks like. As I mentioned, inside they have a muscular foot for burrowing, very similar if you've ever seen a saltwater clam. That's mainly what that tissue is inside of the mussel. They'll stick that outside the shell, kind of burrow down into the stream and river bottom, almost moving like a snail would, creeping along to bury themselves down in the bottom in the substrate. Inside of the shell, they have these teeth that they use to kind of hold the shell together. Lateral teeth and these pseudocardinal teeth. Some species have no teeth. If you ever look at an identification key, that's usually the first couplet in the key is, does the shell have teeth or not? But obviously, if you're looking at a live animal, you're not going to know that because typically you can't tell that unless you pry it open, which you would kill it. So a lot of those keys are based on shell morphology. Yep. Yeah, I see that. It's falling down there. Let me get that. There. Let's tighten that up. There. Hopefully that stays. Typical size is about 30 millimeters to about 100 millimeters in size, so you're looking at a few inches. Some get as large as about a dinner plate, like a washboard mussel. They can get fairly large. Many shapes and sizes. Most shells are thicker, but some are very thin. And then you look at this shell here. This exterior is called the periostracum. The interior is the nacre or the mother of pearl. And then you got some different features on the exterior, like these bumps that we call pustules or a sulcus sometimes. Some are smooth, some are rough. And I'll show you a photograph of just some various live mussels as to what they look like. This is a pile of mussels from Southwest Virginia. Typically when you see them down there, they're fairly distinctive. A lot of different shapes, colors, distinguishing features on the exterior of the shell. Let me go back real quick. So you see some rays on these guys here, sulcus, bumps, different shapes, different colors. Pretty easy to tell some of these apart when you're down in Southwest Virginia. However, when you get to this part of the state, kind of east of the New River, this is a lot of what the mussels look like in this part of the world. These are all different species, but you can see they all look very similar to each other. Kind of a drab brown color, similar shape. That's why they can be pretty hard to identify. A little bit on the ecology. They do have separate sexes, males and females typically. There are some species that are hermaphrodites, but they typically are males and females. And when they spawn, males will release the sperm into the water column, females will siphon it in. It's just kind of a passive thing that happens out in the wild. Some of these guys are fairly long-lived. They can live over 100 years old. We typically age them like you would a tree. You can thin section the shells and look at the growth rings. Each year they usually put on a growth ring, and in the winter they'll stop growing. You can sometimes read that on the exterior of the shell, but usually you got to thin section the shell and look at that just like you would a tree. And sometimes we can tell how old it is based on the size as well. I mentioned they're filter feeders, primarily feeding on phytoplankton, bacteria, detritus, various things in the water column. They're gill breathers. These guys have 4 gills inside the shell, so similar to a fish. They're taking in that water to get their oxygen. Habitat ranges from rivers to creeks, lakes, ponds, pretty much anything that's perennial they can live in. They can't live in intermittent or ephemeral streams. And typically they're not going to be found in these headwater cold streams either. It's usually a lack of food and a lack of the fish that they'll need to complete their life cycle, which I'll mention here shortly. And they're typically considered for pollution tolerance, intolerant to moderate. If you're looking at doing like when people go out and do the SOS surveys with the aquatic invertebrates, if they do pick up mussels, they kind of range as to what their tolerance level is. So here's an example of where you may find them. So it can range from a headwater stream into a typical Piedmont stream down into the coastal plain of Virginia. Some of these typical swamps here, low DO, almost no flow in these. You can have mussels living in there. And then larger rivers like the James River. So like I said, wide range of habitats that these guys can live in. And getting into their life history, their life cycle, basically what they need to do, and which is what is unique with these guys, and I'll get into some of the videos with these in a minute, is you have your female and a male here. You have fertilization occur in the water column. A male will release the sperm. Female will siphon that in, fertilizes the eggs. The eggs will develop into a larvae that we call a Glochidia, which is this guy right here. And that Glochidia is parasitic on typically freshwater fish. It has to get on those fish to complete its life cycle. Not all species do this. Some other species can use organisms like salamanders, but they typically have to get on some kind of a host to complete their life cycle. And once they're on that fish, if they're on the right fish, they will insist on the fish. Usually they're on the gills, but they can also get on the body and the fins. They'll transform into a juvenile. Basically looks the same as the Glochidia, except they got some internal tissue at that point, and they drop off the fish. And hopefully they're in the right habitat, and then they can grow from a juvenile into that adult and continue the cycle. And so there's some strategies when they're trying to use, when they're trying to get on these hosts, where it can be active, where the mussels are physically trying to get the fish to come to them, and then there's these passive delivery where they just kind of broadcast those Glochidia out into the water column. And it's these active species that have kind of the real cool video that I'll show you here in a second. And so they may have this lure. They've got a tissue on the inside of the shell that they may have developed into this lure that mimics maybe a fish that they stick out there to try to get a predator fish to try to come and strike at it. Or they can release these packets of their larvae out in the wild that might look like insect larvae or larval fish that another fish will eat, mistakenly thinking it's a prey item. Or as I mentioned, they can just broadcast release the Glochidia out into the water column. There's one species that releases them out in a mucus web, almost like a spider web, and they use anadromous fish. And so when the fish are running up the river, these Glochidia are dispersed in the water column. The fish swims through them. They latch onto the body, the gills, and the fins. But the fish doesn't know they're there, so that's more of the passive delivery system. These are just some photographs before we get into the video. There's like these displays you can see. This is like a Venus flytrap here. This is the mantle tissue in the lure. Kind of looks like a fish, got the eye spot here. This one's sticking out a lot of mantle tissue with these papillae. It will move the entire shell and look like a crayfish. And then this one here is like fishing for another fish. It has its whole basically charged inside the gill. These are all the larvae that are packaged up into what we call the superconglutinate, and it hangs it out on like a mucus strand, waiting almost like actively fishing for a fish. And then these are the smaller conglutinates here. These are the gills of this particular mussel, the kidney shell. You can see they kind of look like a larval fish. They've got eye spots. They'll release them out, and they kind of just stick on the stream bottom, and a fish will come along thinking that that's a prey item for it. And so, Chris, if you could hit the video for these. So this is the mantle display right here, and that's the gill that's sticking out. In hopes of a bass or a sunfish come and strike at this, that female will then release larvae out into the face of that bass to get them onto the gills. So you can kind of see how, you can see the eye spot there. You can see some striping on there. So it's trying to mimic a fish is what it's doing. It will sit here and it will just wave and wave and wave until a bass or a sunfish or something comes up and starts poking at it. And on the gills, you can see they've got these water tubes just filled with these larvae. And mussel like this can have 10 to hundreds of thousands of those larvae inside there. Those larvae are pretty small. They're like the size of a grain of salt. All right, next one, Chris. This one's like a Venus flytrap. This is a snuff box. Like I said, these mussels live on the bottom, so there's certain fish that come along and flip rocks and sticks looking for aquatic insects and may come across some mussels like this, and you'll see what will happen when one does. So this is a log perch. Thinks it's going to flip that rock over, but it's not a rock. It's a mussel. Now it's grabbed its snout, and what it will do is it will hold on to it and release its larvae right onto its face to get those to get them onto the gills of that fish. This is the one that looks like a crayfish. So that's the whole shell that is out of the water right there. There's the papillae, almost like an eye spot, so it kind of looks like a crayfish as it's moving there, trying to get a fish to come in, like a bass or something like that, to strike at it. And this is the conglutinate. Watch when it spits it out. You'll see little white things. So those are the Glochidia right there, the larvae. But when it spits it out, they go all across the gills, and those Glochidia will snap on the gills. And I'll show you some photos of the Glochidia. They look like a little Pac-Man is what they look like. And they're triggered. They have a chemosensory, so when they hit that gill, they're triggered, and they'll snap shut onto that gill. And this is the last video. This is the one that's got the entire gill cartridge, basically, and it's on a mucus membrane, and you can see that kind of right here. And it's hanging that out there, waiting for a fish to come along and eat that and get all those Glochidia onto it. And so there's lots of other videos like this. If you Google freshwater mussel lures, you can just see all different kinds of these. So these are what the Glochidia look like, just a few types. I said they range in size. Bigger ones are about a third of a millimeter in size, down to less than a tenth of a millimeter. So when you have them in a dish, they do look like grains of sand or salt. Some look like kind of like a hot air balloon or teardrop shaped. Some are a little bit more elongate, and some have these teeth or hooks on the end of them. Those ones are able to get onto the body and fins of fish, whereas these ones typically are more onto the gills. And that's what it looks like when they are on the gills. They'll insist into the gill there. And just because the fish is a proper host doesn't mean that all the Glochidia will stay on that fish. The fish will have an immune response and reject some of those Glochidia. And if the fish isn't a proper host, they'll reject all of them. Some mussels are generalists. They could use a lot of different fish species. Some are specialists. They'll only use one to a few different fish species. And that's one of the reasons why some of the mussels are so rare, and I'll mention that here shortly. And this is a juvenile mussel here. We've got the little foot sticking out, so you can see it looks basically like what the Glochidia did, but it's got the foot. And it uses that initially when it drops off. They pedal feed until the gills fully develop and it starts filter feeding. So why are freshwater mussels important? Why should we care about them? They're great indicators of water quality. They live permanently in water. They're siphoning that water across almost 24/7, 365. So if there's any pollutants in the water, they're going to be impacted by that. And some of these constituent and pollutants in the water, they're fairly sensitive to, more so than any other organisms. Some water quality standards are set based on the impacts of freshwater mussels. Like ammonia, a new water quality standard was set based on toxicity testing that we were able to do with freshwater mussels. They used to use rainbow trout and fathead minnows, but they found that freshwater mussels were much, much more sensitive to ammonia than those organisms were, and now the new water quality standards uses the limits for freshwater mussels if we have those in the streams. And they're sessile, so they're not going to move out of an area like a fish would. So if there's an impact there, they're going to feel it. A fish can go somewhere else, a crayfish can go somewhere else. These guys cannot. So it'd be like, might be like analogous to putting you into a room of smoke or something like that where you couldn't get out and you're constantly breathing that across. That's what happens to these guys if they're stuck at a place and there's some kind of impact going on at that site. Heavy metal, mercury like they have in the South River, silt that would make the water turbid, things of that nature. And so they're great filters. I get a number of calls every year from citizens interested in releasing these in local waterways to clean up the water because they do filter water upwards of about a half to a gallon of water an hour, depending on how active they are. And so you'll see what these guys can do. This is a demonstration we usually do on the stream side when we have activities going on. There's an aquarium here with no mussels and an aquarium with mussels, and you can see after about an hour what starts to happen. You can start to see the mussels, and then after a couple hours you can see how crystal clear the water is on this side versus this side. But I do caution that the mussels can't necessarily clean up the problem. They can help, but sometimes the problem is so overwhelming that they can't do it by themselves, and that's what we're trying to get across to folks. We do tend to tout this ability of freshwater mussels as an ecosystem service, that they do filter and clean water, but they can only do it to a certain point. There may be other remedies that have to go into effect along with releasing freshwater mussels, and if you don't take those remedies, then these guys won't survive when you release them out there. So they're not the panacea to all of the water quality problems that we have. So this is an example of what small mussel bed can do. All these flags are a mussel that were, when we surveyed, they're just marking a mussel. This particular mussel, the rainbow mussels, filters about a half a gallon of water an hour. 575 flags were in this area. And what we typically see is not what's all there. Some of these mussels will be buried below the surface. So we're estimating maybe we're seeing half of what's there. And so you can kind of see 1,150 total mussels in this small area. They're filtering upwards of 14,000 gallons of water a day. So a pretty important ecosystem service for these guys. It's analogous to what was historically talked about with oysters in the Chesapeake Bay. When oysters were in their heyday, they used to filter that bay probably once every 30 days is what's talked about. Freshwater mussels used to do the same in our freshwater streams and rivers. And so, healthy mussels can give you an idea that the streams and rivers are healthy, and then also relates to our health because we use streams and rivers for different purposes. You may fish and eat the fish out of those systems, drinking water, and then different recreational activities. So it's that concept that everything is tied together. They're important food sources for a variety of animals. Mammals like raccoons, muskrats, otters will feed on them quite readily. Some fish and some turtles will eat them at smaller sizes, and then things like blue catfish, which is an invasive species in Virginia, which was introduced by my agency back in the early 70s as a sport fish, and now is kind of a pest out there. We see them eating the mussels on the river bottom, out in some of the rivers like the Mattaponi and places like that out in eastern Virginia. Habitat for other animals and plants. You can see algae growing on this guy here. Mad toms will use the shell to lay their eggs in. Aquatic insects will often, you'll find them on the shells of the mussels. It's not uncommon where you have healthy mussel populations to have a higher diversity of fish and aquatic insects at that same location. Historically, Native Americans used these for a number of reasons. One, for food, and then also for utensils, things like bowls, spoons, necklaces. Some were fashioned into tips for spears and things of that nature. As I said, Native Americans did eat these. They were oftentimes, you see these in archaeological digs along the rivers, they were an important source of food for Native Americans. When I'm working, this is probably the most common question I get asked by the general public is, can you eat them? Technically in Virginia, it's illegal to eat them, but what I usually tell folks who I know have eaten them and I ask them what they taste like, they taste like rubbery mud. Because of that muscular foot, it's like chewing a big piece of rubber. If you've ever had saltwater clams, typically they strip them and fry them. So it's like chewing a big rubber ball, and if you think if they're sitting in the bottom of a stream or a river, they're sitting in sand, silt, mud, gravel, cobble, they're taking in that. If they've lived in there for 30 or 50 years, that's exactly what they're going to taste like. So I tell someone, unless you're willing to put a straw in there and drink the water out, then I wouldn't eat them. Historically, there was a huge industry for buttons prior to the invention of plastics and zippers. So if anybody has older clothing, they might actually be from mussel shells. They would collect the shells, punch the buttons out, and put them on shirts and dresses and things of that nature. That's obviously gone by the wayside with plastics and zippers. One industry that still exists is cultured pearl industry. It's not as big as it used to be, but what they'll do is they'll collect certain species of shell, punch this bead out of the shell, and they put it between the shell and the tissue of an oyster, and that gives it the start for a freshwater pearl. So a lot of times these freshwater pearls that you'll see will have a mussel shell inside the center. They're able to use plastics more nowadays than they were initially able to do, but some states like Kentucky, Tennessee, Alabama still has a commercial harvest of freshwater mussels for the cultural pearl industry, and typically those shells will be shipped off to places like China where they're used for that. And this is an old photo from 1938. This is a pearl diver, so you can see the length they went to try to get some of these mussels. So he has a converted gasoline tank on his head with a hose that he would use to go down for upwards of an hour to an hour and a half to try to collect mussels. So. Worldwide, there's about 900 species of mussels, and you can kind of see the rough diversity here. The thing that should stand out, that I'll point out, is North America has the highest diversity. We're about 300 species. Next highest would be if you get over into China, India, this area here. But by far, we have the most in North America. And if you kind of drill down and look at those numbers in North America where the highest diversity is, southeast U.S. You can see these numbers here. You get west of this Mississippi, it really peters out to just single digits out here in the plains in western U.S. But you can see over here, Virginia, we have about 81 species. That'll rank us about 6th in the U.S. for number of species. And that's fairly analogous to what you see for other aquatic organisms like reptiles and amphibians and fish and crayfish. A lot of diversity here in southeast United States. And then across Virginia, the highest diversity is in southwest Virginia. Just over 50 species. We have about 30 species across the rest of the state, and we have 41 T&E or threatened and endangered species in the state. 34 of them occur down here in southwest Virginia. So historically, most of the mussel work was done down in this part of the state. So when I did my graduate work, this is where I did it. Most of the work I do now, I'm kind of east of the New River here, just because historically not as much work had been done out there. Doesn't mean the mussels aren't as important, but just because of the resources down in southwest, that's where the limited resources were spent. And their status. You look at how many species are considered stable. 17 out of 81. So it's about 3/4 of the species in Virginia are considered threatened, endangered, extinct, or imperiled at some level. And that mirrors what you typically see on the federal level as well in all the other states. About 3/4 of the species are considered threatened, endangered, extinct, or imperiled. Special concern. So that kind of shows here some of the different taxa groups. The only one that would be above this if we threw freshwater snails on there, that gets right about with mussels at about 80%. But you can see other organisms that get much more attention, like mammals and birds. They're down at the bottom of the list here. So it's kind of an inverse of where the resources go for conservation activities for some of these organisms. And so why are they so imperiled? What are the threats? Then I'll mention some of those. There's a whole host of threats. You guys are familiar here in Waynesboro with the DuPont plant and the mercury, so that's one example, heavy metals. You got potential for exotic species. These are zebra mussels. Fortunately, we don't have those in Virginia, but across the Mississippi, Midwest, down to the Gulf Coast, you get these guys. And you can see for a filter feeder, it's not the best thing in the world when you have a bunch of other critters latched onto your shell, not allowing you to open to do your thing. So that, you can see where that would be a problem. Impoundments or dams changes the habitat upstream and downstream, changes the flow, basically changes the whole dynamic of the river through here. And also creates a barrier. So if they're using fish as a host, those fish move up and down the system. Most fish are not going to get past that barrier there. So if you lose animals upstream here and they're still downstream, they can't recolonize up there. You get discharges. You have a lot of these across the state from water treatment plant or wastewater treatment plants. And then you get these random chemical spills. This is where I did a lot of my graduate work right here. Actually, this happened in 1998 down in Southwest Virginia. Tanker truck made a turn too fast, spilled a rubber accelerant. It went right down a drain into the Clinch River, wiped out 7 miles of the river. It's the biggest T&E kill on record for Fish and Wildlife Service. Basically, from the town of Cedar Bluff down to Richlands, it killed everything in the river. And so these things happen more than you would think, and they happen in the wrong places. This is usually what happens, where you have some of the rare things. As we develop the landscape, you get lack of best management practices, so you get sedimentation where it can be prevented. You get forest fragmentation, so you lose riparian buffer, so you don't have that shade around the stream. You don't have that leaf input for potential for food source for the mussels. You don't have that filtering capacity for filter out pollutants coming across the landscape. This is probably one of the biggest things, is just loss of that riparian buffer across the landscape. And this is what you tend to see as that happens, sediment just coming into the rivers. So I mentioned earlier, analogous to this would be a human sitting in a closed room with a lot of smoke and you're breathing across and it's irritating your lungs. Mussel can't go anywhere, so it either has to close up and ride this out, or it takes that across its gills constantly. Not a good scenario. And so you see this a lot on rivers out there where the bank is just eroding and it's dumping in tons of sediment at locations like this. And so then that sediment settles out somewhere and it smothers the bottom. So if you can't get out of the way, it's like putting a blanket over top of some of these mussels. Fish, they can hopefully go somewhere else. Mussels, not so much. So what are we trying to do? What are our restoration options for freshwater mussels? Sometimes it's something as simple with working with the landowner and doing best management practices, where it may be fencing livestock out of a stream. That's a real easy, simple solution. Usually doesn't cost a whole lot of money. And you can see an example of that here. There's an area that wasn't fenced out, and then after a couple years, you can see how that's kind of grown back. And sometimes that's all you need to do. You don't need to do anything. It's not necessarily rocket science. You don't have to come in here and spend a lot of money to restore this area. It's just giving those livestock an alternate source of water somewhere so they're not going down the river. And this is a lot of what we try to do when we're trying to restore habitat. But sometimes that's not enough. Sometimes you actually have to get in there and do stream restoration and kind of stabilize these banks that are eroding. If you're ever driving across Virginia and cross streams, I look at them all the time when I cross them on a bridge. This is typically what you may see. This is probably a hay field. There's no vegetation except this short-rooted grass. As the landscape develops and more flow comes into these streams and rivers, the hydrology changes, you have more hard surfaces. The banks just can't handle it. So it starts to erode and widen out and deepen because you don't have any root material holding that. So what we try to do is come in and grade that back and plant that so it holds over time to reduce that sedimentation. And sometimes they're so bad across the landscape you can't fix anything with it. The bigger thing we're trying to do, and hopefully if you have the habitat and the good water quality, is to do freshwater mussel propagation. And that's part of what I'll talk about with the South River project. And this is an example here. So this is what we're trying to do, is to get large numbers of freshwater mussels at size, maybe 1 to 2 to 3 years old, and release them back at the sites where they're either are no longer at, but conditions look like they're suitable, or where they're at, but the mussels just struggle to recover. Because if you think about that life cycle they have and how long-lived they are, they're typically not an organism that responds quickly. Aquatic insects can come back into an area quickly, like that chemical spill I mentioned. Aquatic insects are back in there pretty thick. Freshwater mussels, not so much. If the fish aren't there, the freshwater mussels are not going to be there either. But even if the fish are there, because of that life cycle, and it takes them a while to get the size, a while to become reproductively mature, and then continue to have individuals come into a population, it may take decades for them to recover. So we're trying to speed that up by propagating them at hatcheries and releasing them out. It's similar to what they do with fish. But we're not nearly as far along as the folks with fish. They've been doing freshwater fish culture for 100 years. We've basically been doing freshwater mussel culture. It started in the early 1900s, then there was a long gap where it wasn't going on. It's been more in the past 3 decades is what's been going on, so we still have a lot to learn. And so this is what we hope to see, and this is what we want to see and what we are seeing with the South River project. Lots of mussels back out on the landscape. And so in Virginia, we have 3 facilities that are doing this work. The first one came online in the mid-90s at Virginia Tech, the Freshwater Mussels Conservation Center. This is where Jess Jones works. This was basically kind of started up when I was a graduate student at Virginia Tech back from 95 to 98. And this is just kind of an overview. This is the general aquaculture facility out there. They've got some buildings with the pond where they've got systems in some of these buildings where they do the work. In 1998, Department of Wildlife Resources started a facility in Southwest Virginia, Marion, Virginia, down at our Buller Fish Hatchery to do the same thing Virginia Tech was doing. Basically, these are 2 kind of big sheds on top of abandoned raceways. So it's not high-tech, fancy buildings that some of this is going on. It's usually kind of repurposed structures that some of this goes on in. And so this started in 98, and over that time, almost 7 million mussels have been produced, and they've released over 140,000. And that's mainly been down in Southwest Virginia. The focus, as I showed you before, most of the mussels are down there, most of the T&E species are there, so Virginia Tech and AWCC did most of their work down in Southwest Virginia. Fast forward to 2007, we started a cooperative facility with U.S. Fish and Wildlife Service in Southeast Virginia at the Harrison Lake National Fish Hatchery to focus on species in Eastern Virginia because they weren't getting the attention that the Southwest Virginia mussels were. And so I helped start this up with the hatchery manager there at the time, and this is the facility that's working with the one at Virginia Tech to do the work for the South River project. And over those 14 years, we've produced about 13.6 million mussels, and we've released over 312,000. And so you can kind of see, well, you produced a lot, but the numbers released aren't nearly as high. Typically, if you're getting 5, 10, 15% survival of what you produce, released back out, that's usually a good number. At some point, we weren't even able to grow these guys past a few weeks. And so over time, we've gotten them to be able to grow them to a few months to a year to almost hold them indefinitely. And when we first started, we released things that were just a few weeks old and we weren't seeing any return for our results. Now we'll raise these mussels so we can actually put a tag on them so they're a year or 2 old And we see much better results than we did for probably the first 10 years when we were doing this work at Virginia Tech and down at AWCC in Southwest Virginia. It's night and day difference now than it was 20 years ago. And so how do we do this? Basically, we got to go out and get the female mussels that have the larvae. Got to extract the larvae out of those. We check to make sure the larvae are viable. You got to have the host fish, and then you basically introduce the larvae, the Glochidia, and the host fish together. As I said, some of this is pretty simple. Sometimes you put them in buckets or containers together for 30 to 60 minutes and just let the Glochidia attach to the fish. It's, like I said, it's not rocket science, some of this stuff. Some of it's pretty basic. Trying to get them to size is a little bit more difficult, but actually doing the process is fairly simple. You put those fish into various systems, might be aquariums, might be larger tanks. You wait for those larvae to develop and drop off, and you collect them, and then you put them into various grow-out systems that we've developed over the past 2 to 3 decades. And so that's the hope, and then once you get them into those, you hope to have them a year or two and then get them back out into the rivers. And so the goal, as I mentioned, is to either augment or increase existing populations, expand existing populations into areas where native mussels may be able to get but are having a hard time doing, or you reintroduce them into areas that they can't physically get to anymore. And we're doing some of all of these with the South River project. So the South River still does have some mussels in it, but you have to get above Waynesboro where the mercury impact is. So we are releasing some mussels there to increase numbers, but we're also releasing mussels in areas where they're currently not at. So we're doing kind of a three-pronged approach. And where have we worked across the state? Everything is green. These are watersheds, major watersheds. Everything in green, we've worked somewhere in there for doing mussel propagation and release. So you got the James here, we've worked across, the Roanoke down in the Dan system. There was a coal ash spill there in 2014. We've done a lot of work there. The Chowan system, here we are in the Shenandoah and the Potomac, we've done work. The Rappahannock and the York. The only systems we haven't really done actual release work is this little portion of the Yadkin and then the Big Sandy up here. This is kind of coal country in Virginia up here. Not a lot of mussels up there, not a lot down here, not a whole lot in the new. Used to be a lot more, but we're trying to get into the new to do some recovery as well. And so the DuPont South River project, this is just a little kind of a timeline of the situation, and most of you are probably familiar with that, so I'm not going to get into detail on that, but basically you've got mercury being released from the late 1920s to present, and then various things happened in the area as far as fish consumptions, the South River Science Team being formed to study the problem, and then the NERDAR case, which is the Natural Resource Damage Assessment and Restoration. That was when the Fish and Wildlife Service basically, lack of a better word, would sue DuPont and work with them to try to come up with restoration activities for the damages from the old DuPont plant. And that settlement was approved in 2017. That was a $42 million plus settlement for various activities within the South River watershed. And so that covered basically the South River up in the headwaters above Waynesboro down through the South Fork of the Shenandoah, even into the main stem of the Shenandoah River. So this is kind of the coverage area that we're looking to do restoration in with freshwater mussels for the South River DuPont project. And so we had $4 million for restoration. Project we were looking at taking probably about 10 years. It'll probably last longer than that. And the goal was to release at least 280,000 mussels of 9 species over those 10 years. So you're looking at about 28,000 mussels a year or 14,000 mussels per facility. That's the goal. That's like the benchmark, the minimum we're trying to hit. If we get over that, great, but that's kind of the target where we're trying to go for. And what's the potential benefits of doing this? And this gets into some of the things I'd mentioned about why mussels were important. You know, mussels, if you have a healthy bed, providing cleaner water, they can stabilize the river bottom. You have a lot of them there, they hold things together. Increase food abundance for other organisms. You know, they're kind of the bottom of that food chain for some of these organisms. So more mussels, it helps with some of the higher level organisms as well. And so where have we worked so far? 5 sites we've released mussels, and we started this project initially kind of in an infancy in 2018, but really in 2019 is when we really started doing propagation at both facilities. And so this is, let's see, you got, here's Waynesboro here, Stanton, Harrisonburg, Charlottesville. So these are 3 sites on the South River, Stewart's Draft, this is Lyndhurst, and then this is Ridgeview Park. So the event we had on the 12th of October, that was at the park there. And then down in the South Fork Shenandoah, a couple spots down here in Warren and Page counties, Foster's Boat Ramp, and then this is Hazard Mill Rec area right here. So those are the 5 sites we've worked at so far. Those aren't going to be the only sites we work at, but that's where we started so far, to release the mussels and then monitor them to make sure they're surviving over time. And so what have we released so far? And these are the numbers and the different species. We've done 6 species so far of the 9. You can see which ones we've released in the South River, which ones in the South Fork Shenandoah. We're getting upwards of about 15,000 total. So you can see our goal was 28,000 a year. So we're definitely behind, but typically it's usually slower on the front end because you got to kind of build up, learn what you're doing with some of these species. And also it takes a couple years to grow the species the size to release. So our production numbers at the 2 facilities, this is the one down in Southeast Virginia, this is the one at Virginia Tech. You can see what we've produced. So at both facilities, we have tens of thousands of mussels sitting there waiting to be released that are ready now that will be releasing over the next year or 2. So these numbers will jump up quite a bit over the next couple years, and if we continue to produce like that, those numbers will raise up, and we'll have many more than 5 sites out in the landscape and many more mussels at these sites as well. And this species here, this is the brook floater. This is kind of the big deal for me with this project. It's a state endangered species in Virginia, believed to be extirpated from Virginia. The last time it was seen live was in 1998 up near a stream by Manassas. It's kind of its hub in Virginia was the Potomac and the Shenandoah Basin. So in the Potomac, Northern Virginia, the impacts are development. In Shenandoah, it's agriculture. It's heavy agriculture. All the ag is right next to the rivers, tight valley, water quality issues. So we've released this one at all 3 sites in the South River and 1 site in the South Fork Shenandoah. And they've been in there, some of them have been in there for a couple of years, and we've gone back this year to monitor them, and some of them look fairly good. So these are the only known populations we have of this species now in Virginia. So it's considered a restored species as long as it stays there. And tracking of these guys, we do this by various means. This is just like a flexible plastic tag you can glue on these guys with a unique number. It identifies each individual, so we can come back and collect that and see how much it's grown over time, see if it's a female, if it's reproducing. Those are 2 of our benchmarks. Are they surviving? Are they reproducing? And then also, are they recruiting new individuals to the population? That's the ultimate goal. We don't want to have to continue to be putting mussels out like a put-and-take fishery. We want to put them out there over the next 10 to 12 years, have them sustain themselves, and then go on to another area. This is just a rubber hardened black super glue. When we're kind of batch tagging mussels, huge numbers, we'll use this, and you can see some of the techs get creative in how they tag these guys. When you're tagging thousands or tens of thousands of mussels, it can get a little bit tedious at times, so they kind of try to humor themselves. Laser engraver, this allows us to tag these guys a lot faster, and this is basically a piece of equipment that they use for making plaques and trophies. Same thing. So we'll etch the number into the shell. It goes down in the periostracum. You don't want to go too deep. You go too deep, you start to hit the tissue on the other side and can kill them. But This allows us to tag a lot of mussels pretty quickly. So we can have that unique identifier on them. And then we will tag mussels also with these pit tags. These give off a signal. And so we can come out with like this metal detector and look for these guys. Because as I mentioned earlier, not all mussels are at the surface. These guys can be really hard to find. Even I've been doing this for 25 years. Those of us who are experienced are not really good at finding these. Our detection level, if we're good, is like 30 to 40% of what we know is there. And so sometimes, certain times of the year, 1 week to another, a lot of these will be subsurface and you just will not physically be able to see them unless you're digging and digging in the substrate, which you usually don't want to do because you're disturbing that. So you can come in with this antenna here and you can scan it across the bottom and you could ping a mussel. And then you could try to recover it and see if it's alive and get data off of it, or you can just record that and assume it's alive. Sometimes even when we ping these guys and we know the area that they may be in, about the size of this desk, we still can't find them. They may be burrowed down 12, 14, 16, 18 inches below the surface. And so you're digging. I've spent a half hour at a spot and not found the mussel that was pinged. Sometimes we'll find another mussel that we had no idea was there and not the one we're looking for, but we never find the one we're looking for. So those are just some of the ways that we're monitoring those mussels that were going out on the site. And when we did the release on the 12th of October, we had mussels that were pit tagged like this, and then we had other ones that were laser engraved and some that had the that plastic tag. It's a Hall print tag, we call it. That's the name of the company out of Australia that makes it. Some of the more sensitive mussels, we will avoid laser engraving just in case they have any impacts. We'll glue that tag on there. But yeah, and so we haven't done a lot of monitoring yet. We've done a little bit. Like I said, these guys are long-lived. It may take 5, 10 years to see kind of the fruits of your labor out there as far as new recruits coming in. So right now what we're looking at is, are they surviving? What's the survival rate out there? So far it's been pretty high from what we've seen. You're never going to get 100%. But then also the next phase is, as these grow, are they reproducing out in the wild? Are you seeing those females that are gravid? And then from there, you're going to have that lag to when you might see some other individuals that aren't tagged. That will give us an indication that those are the ones we put out that are then recruiting into a population. Now, if you're augmenting a population where mussels already there, you may not know if it's your mussels versus the one that's already there. But if you're putting them into a spot where they're not known from, then you know any new recruits are coming from mussels that you put out there. And so, we hope to work in the South River, hopefully for the next probably 10 years. I said it was initially a 10-year project, but it all depends on how fast we spend that $4 million. It's probably going to be more like a 12 to 14-year project. And we keep cranking those mussels out into the stream. And we probably will put some below Waynesboro. I'm curious to see if they will do okay where the mercury is a bit higher. We did a survey right at the park, downstream, immediately downstream of the old plant. Where there's some restoration work going to go on. We had to verify that there were no mussels there. The habitat doesn't look too bad near the railroad trestle where the covered area is on the walkway there. We saw lots of snails, lots of fish. Habitat looked good. So with one of our common species, we probably will put some out there. They may be out in the stream bottom. We may put them into some cages to be able to collect them over time and do some testing on them to see if they're picking up the heavy metals in their tissue. But the hope would be to get them into that stretch of the river as well, as long as they're able to survive and reproduce, and not just do them above Waynesboro and then go so far down in the South Fork Shenandoah River that you still have this huge, huge gap in where the mussels are occupying. And that was all I had for you guys.
Any questions? All right, over here. I'll start at the back and work my way forward. We use the mic so that the live stream visitors can hear the question clearly. So,
Thank you.
The host fish for the mussels. Are there plenty of those available where you're seeding the mussels? You've got plenty of host fish, or is there any thought to seeding host fish for the mussels?
Are you talking for the South River project or just in general for?
In general?
Okay, yeah, typically we will look to see if the host fish are there, because obviously if you don't have the host fish there, long term the mussels are not going to continue to recruit into the population. We do have some species down in Southwest Virginia where the host fish is no longer present. Dams, TVA dams down in Tennessee has kind of eliminated them either completely, or the numbers are so small that they're just not there. And there has been a thought of, can we restore some of these fish species? Honestly, with the number of mussel species we have and the limited resources, we're not at that point to where if we're going to restore that mussel, we're able to put in the time and the effort to restore the fish as well. But it has been something we've talked about with some of these species, because it would be a two-pronged restoration approach. You would have to do the mussels and the fish, or if you do the mussels, it's something you're going to have to put them out and kind of continue to replenish them over time. So you still have them there, but they're not going to be able to recruit on their own. And so that's where it almost gets into like a put-and-take fishery, where you put them in, and because they're so long-lived, they'll be able to survive out there for decades, but you'll have to come in and supplement them every 5 or 10 years as your individuals are dying out.
First off, I want to thank you for your work in helping the populations of the mussels. I was wondering what kinds of things do the mussels filter out of the water, and what kind of things they don't? And I think I finally understand from your talk that you're helping the populations of the mussels, but it doesn't really help the pollution in the South River. Is that correct?
It can, to a level. I mean, when you have things like heavy metals, like mercury, that mercury is not going away. It doesn't break down like other constituents in the water. So you can see that's been released since the late 20s, and it's still leaching out in places. And so the goal isn't necessarily for the mussels to uptake the mercury. When you have a situation like that, if they're trying to get rid of the mercury, you'd almost have to dredge that out, but then you have impacts with that as well if you're digging up the river bottom. So mussels can take that up in their tissues, but it's not going to be at the level that would be needed to like remediate the mercury issue in the South River. So the thought is more to just restore into healthy places, or into places that look healthy now, where we can bring the mussels back to provide those other ecosystem services that they once did before they were impacted by that mercury. And you ask what they filter. Basically anything that's in the water column. You get sediments, you get detritus, they could take heavy metals out. Like the sediments, they can feed on that to a certain point, but when it's so overwhelming, what they do is they package it up into what they call pseudofeces, and they spit that out, and other organisms on the bottom may eat that if it's not a food item for the freshwater mussels. But like I said, they can only filter to a certain extent. If it's so overwhelming for them, and even if you have a huge number of mussels, if that impact is going to be too much for them, that there's almost no amount you could put in there to correct that without some other type of activity going on along with the mussel restoration. So it is restoring. At one level, it's trying to bring the mussels back because they have been so heavily impacted. And it's also trying to restore the ecosystem services at some level because the mussels have been so depressed. Trying to get them to such large numbers where we can really get those ecosystem services thriving may be a little bit more difficult for this project, but we are trying to do that in places where we have other situations where we do have mussels, but they're just kind of depressed and they can't increase their numbers for whatever reason. Sometimes those are a little bit easier to increase those numbers to a capacity where those ecosystem services are more fully functional. And it also depends on the species you're working with. Some of these species we're working with in the South River, they're not typically really abundant in mussel beds when you see them out in healthy mussel beds. There's like 1 to 2 species that are your dominant, and those will be the most of the individuals that we're going to be releasing, and then we'll seed in some of these other more rare species just to recover them from a rare species standpoint. But other areas we're working, you might have 4 or 5 species where they are really dominant in a bed, and you could really get a large number of those out there really quickly. So we're dealing with a little bit of a different situation here in the South River, and that makes it a little bit more difficult for us to get large numbers out on the landscape really quickly like we can elsewhere.
Quick question on, I'm especially interested, first of all, thank you for your presentation, especially interested in the reproductive cycle. And I wonder if you had to think in terms of roadblocks that would maybe prevent as high an efficiency as you would want to find, would one of them be the larvae attaching to the gills of the fish? And to follow up on that, is that a symbiotic or is it really just a fully parasitic relationship?
I'll answer the second part first. It is
Okay.
parasitic. The fish is getting nothing from the larvae or the Glochidia being on them. So at one time it was thought that it was just a travel mechanism that was the way the mussels moved around, because obviously they're sedentary. They're not going to move upstream. They might move from me to you upstream, but they're not going to get miles upstream. So that's how they moved around. But they are getting nutrients off of that fish. It is a truly parasitic relationship because we can grow mussels. I mentioned one way, the typical way we do that when we introduce the host fish and the larvae together. You can also transform those larvae in a petri dish with the proper media and blood serum. And the blood serum is what's critical there typically because they're getting the nutrients out of that. You might have to use carp, horse, rabbit. There's different serums you can use. So they're definitely getting something out of that fish. And that is a barrier. That's one of the reasons why they are so imperiled. Besides them being sedentary, not being able to get out of the way of impacts, but you think of that complex life cycle. If they were able to skip that like some other mollusks do, like we have an invasive clam, the Asian clam, they don't use host fish. They reproduce in a similar means, but they don't have that host fish. They can do well. They get into an area and they can just boom in numbers. So if they were able to skip that, but then they don't have the mechanism to move around like some of these other species, so they still need that. Now, there are some species that can skip the host fish. They will transform directly in the gills. They'll be the juveniles and they'll release them. Surprisingly, one species we work with is for the South River project, the green floater. They're not doing so well. So there's something going on there with those juveniles in the wild that's impacting them, potentially more so than some of these other species that are using host fish. Because you would think if they skip that phase and they're releasing hundreds of thousands of juveniles right out, and if they're in a good area, they would do okay. Not so much. That species is shorter lived. It may live only 10 years, so sometimes you'll see it boom in an area and then disappear pretty quickly.
I have a follow-up. It's a bit personal. My granddaughter is studying mussels in Switzerland, and the tiger mussel, I think it is, and the problem there is mussel infestation around freshwater drinking water, potable water pipes that are drawing water from a lake, for example, to serve a community. They're being blocked, apparently, by masses of zebra mussels. That's probably a story you've heard before, but there's a downside to mussel growth, too, I suppose.
Yeah, zebra mussels, yes. They're quite a bit different than the native mussels we're working with, like zebra mussels, and there are some other native mussels, like brackish mussels in the United States, that they are more colonial. They have these byssal threads, these hairy structures that they use to mass and build up. You can get zebra mussels. They'll just continue as long as they got something stable to start on. They just continue to build. These freshwater mussels, they're solitary, so they're not going to form like a colony like a zebra mussel would. They do have a byssal thread, that hairy structure when they're smaller. It's usually a single thread. And they usually use that to attach either to a substrate or they might attach to another mussel to kind of anchor themselves when they're small. And as they get larger, that disappears. They don't have that. So when you have the freshwater mussels, they're not attracted to, say, like an intake where they're going to colonize that and attach to it. They don't have that byssal thread structure to be able to do that. The zebra mussels do. That's why there's such a financial impact. When you saw them in the Great Lakes first get in there, they shut down the water supply for a town, I think it was in Michigan, because they didn't realize they were clogging their pipes, and then it clogged their pipe, and they were like shut down for a week, I believe it was, until they got in there and cleaned them out. But that's why it's such a huge financial burden for some of these industries where they do have intakes. They got to continually treat them and clean them out because there's no other way. Once those zebra mussels were out there, they're not getting rid of them. And we're fortunate not to have them in Virginia. We did have them at one site. It was up in a quarry in northern Virginia. Right when I started, they found that population up there, and we were able to eradicate it. I still think it's the only eradication of a zebra mussel population that's happened, but the situation was just perfect where it was in the quarry. It was isolated from any kind of flowing water body, so we were able to treat it with actually commercial grade fertilizer. Mussels are very sensitive to potassium, very sensitive to potassium. So we basically fertilized the heck out of the quarry, and they couldn't sense it like other things. Like with chlorine and things, they'll close up and try to ride it out. They could not sense it, so as they continued to filter it across, it breaks their gills down. And so that's what happened with them there.
There are no biological remedies, a bacterium that would love to get the zebra mussel?
They've been looking at that. That was going on back when we were looking at potential options for eradicating the zebra mussels from that quarry up in northern Virginia back in 2002, and I know that's still being looked at as far as introducing that into some kind of particulates that the zebra mussels will then filter through, and it will only impact them. That's usually the hard thing, is to get it something so focused that it only impacts that organism and not other organisms. And I think there's one out there. I haven't been following it that closely recently, but I haven't seen it be used anywhere else. The potassium option was used out at an Air Force base. Well, actually, I take that back. I think we recommended they use the potassium, and they went with copper sulfate, and they didn't kill everything in the lake they had out there on the Air Force base, and the zebra mussels ended up coming back.
Questions about the South River. Is the mussel population really barren below the discharge point at DuPont?
Yes.
And how far downriver does that barren aspect go?
I'm not sure exactly how far down that goes because we haven't done a lot of recent surveys in the South Fork of the Shenandoah. I would say in the South River itself, below the plant, all the way down to the South Fork, Shenandoah.
So as far as Port Republic, it's barren then?
As far as we know. Now, there could be mussels there that are they're so rare that we just can't find them. Because like I mentioned before, sometimes our detection rate isn't the greatest, and it's not necessarily a small stream to go out and sample in, so you could have a relatively low number there and just not be seeing them. We usually look for shell material, but if the shell material is relic, it could have been stuff that died, decades ago that's just hanging out there. The condition can give us an idea if it's more fresh, but sometimes you can tell it's been there for 10, 20, 30 years.
You spoke a little bit about the fact that you're monitoring the river just down at the railroad bridge here in town. Is part of your plan to hopefully be able to establish mussels in the south between where the plant was and Port Republic?
My hope is to at least have one site where we've got them restored. If we're producing enough, it's really how many we produce and then how many we could put out onto the landscape. So if we have limited numbers, then you try to focus more of those into a small number of sites rather than spread them out too thin over a large number of sites. But my hope is to probably put some at that location we saw at the park, downstream of the plant where the habitat looked good and the other organisms looked like they were doing well, and see how they do. And if they do okay, then maybe to move to some other locations downstream of the plant down to Port Republic. The bulk of the work is planned to go further downstream as the mercury lessens, and then upstream of Waynesboro, and then maybe into some other tributaries within the watershed like Christians Creek and places like that. But I have not written off the area below the old DuPont plant. I would like to put some in there and see if they do well, and to recolonize that area if we can. That I'm not certain of. Somebody mentioned Back Creek, and I haven't surveyed in there, so I would have to go look at that and see. Some species are more suitable to smaller streams than others, and so that is a stream that we are going to look at and see if it's suitable for release in the mussels as an additional option to the south and the South Fork Shenandoah.
Any other questions? I have one. Do you have an estimate, or I guess it would be probably just a wild guess, what the survival rate is once you seed these mussels?
Yeah, based on early monitoring, within the past year or two since they've gone out, we've seen survival anywhere from 70, 75%, upwards over 90%. Now the trick is, if you're not finding the mussels, have they survived or have they died and washed off somewhere? So usually on these projects, what we try to do is pit tag at least a subset, 10 to 20%, and look for those to kind of get an idea of what our survival is. But even with those, some of those are going to wash downstream somewhere, and you can find them miles downstream. And so if they're off sites, if you've released 100 mussels at a site and you find 60 of them but have no evidence of the other 40, typically what the analysis will be is that you have 100% survival, but you're unable to find those other 40, and the assumption is they're off somewhere else. You just can't find them. You just can't cover that area. Because you can see with that antenna, we can't cover that long of an area. Now they do make another antenna that floats. You can pull it behind a canoe or something like that, but if a mussel gets washed off, a mile or two downriver, it might be hard to find that. But just based on other projects, if the conditions are right, survival is usually fairly high. We released some brook floater up in Broad Run, which is the site where it was last found live near Manassas, and we found more mussels up there than I have when we've done monitoring. And I think we found like 82 of the pit tagged ones out of 110. So pretty high recovery rate. And my guess would be, there's probably some of those that died, but there's probably some that are off site as well. So if you can get 80 to 90% survival with the numbers we're trying to release, that's usually pretty good.
Wow. Well, I'll tell you, when Dr. Jones came and did the talk on mussels a couple years ago, I thought that was one of the best programs, and you didn't disappoint today.
Thank you.
This was a great program and very exciting, and I learned something new every time, and mussels are just fascinating, and I never had any idea how fascinating it was. So thank you, Brian, for a great presentation, and thank you for all coming out tonight, and hope to see you January 11th for our next Science Talk. Have a great holiday.
Thank you.